And just to touch a little bit more, you have nematodes as well, which is another important, what we call super predator in the soil system. And let's touch a little bit on diatoms. So diatoms can not only generate energy through minerals in the water, which is generally how they do it, but they also have a chloroplast. So when you look at a live one, you can actually see the little photocell that's using sunlight. So these guys have two energy sources. So a very, very important thing to understand that they can thrive and survive in completely different environments, void of light and within light. A lot of biology can't do that. And secondly, their silica is biologically derived, which means that it's a lot more biologically active or available for microbes to eat or bacteria to consume. Why is that important? Well, if the bacteria consumes a dead diatom, it's going to store that silica in a plant available form within its body. As long as comes a protozoa, it eats that bacteria, and all it cares for is the carbon. So all of that other stuff is released. So recently, Keith sent me a paper talking about micronizing or nanoparticles of silica being used in sodic soils, so high in salt, which has always been a problem. If you have too much salt, it really shuts down a lot of the ability for that plant to function or even get close to producing the things that you're expecting it to do in agriculture. So to have that as another tool in the toolbox for conventional farming that you're trying to transition an existing field is huge. I mean, it's incredible that you have access to these types of tools, thanks to Keith. And another important thing to understand too is that we know for sure that fungi, bacteria, and plant roots all need oxygen. So in a compacted soil, how do you start? Where do you start? You go out and spray a bunch of product on top of the surface, and it's not going to penetrate. It's not going to get down into the soil system. This is why you need cover crops, so that you can have that root aggregate or the soil aggregates to allow stuff to penetrate down in. And another thing that Keith has not mentioned here too is that I've had no problems turning a compacted, traditional, conventional ag field by using fish brew, but not aerobically stabilizing it 100%. In other words, what I'm looking for is a group called facultative anaerobes and anaerobic bacteria, which can live in no oxygen in compacted soils. By using them, it's kind of like sending in the Marines. You loosen everything up so that then when you do apply a full aerobic fish brew product, you are going to get incredible results because you've softened the ground up to accept this. So that's just some of the work that I've been doing over the last 12 years, whatever it's been, is understanding that, you know, you really need to start at a point, not just throw the kitchen sink at it, go about it methodically. So, and again, we have not spent time really getting into the weeds with taking Keith's product and stabilizing it only so far. But that this is something that can be done at home. As he said, you know, with the diatom levels, you know, this product has shelf stability. 3 years is incredible. No one's been able to do that, not without a pH suspension. So in the future, it'll be something that, you know, once he gets through this next year or so and really has all of these things under his belt, because he's been in build mode. I don't know how long, dude, 10 years?
Yeah, well, 7 years.
7. So he's going to catch up eventually. And when he does, then we'll go down that rabbit hole of figuring out how best to provide what I want to call the startup product that gets you there quicker. Now, that being said, it's not that this doesn't work, because it does. And we're going to have Kyle on here in another 45 minutes discussing the fact that he just applied it and it worked. And it worked to a level that is undeniable based on all the testing we did.